we're going to start the next session. My name is Ana Mellado. I'm part of the LifeWatch ERIC team, and I'm going to chair the last session of the day, which is focused on two main topics: ecosystem and habitat mapping, and natural capital and the One Health approach. First of all, I would like to remind the 6 speakers of this session that you have 12 minutes for your presentation, and we will have 3 minutes for questions from the audience. I would like to welcome our first speaker, Fernando Rodriguez. Fernando is Professor of Applied Economics at the University of Salamanca and Dean of the Faculty of Agricultural and Environmental Sciences of this same university. Fernando, please, the floor is yours.
Thank you so much. Thank you for the presentation. Thank you for the invitation. It's a pleasure for me to be here with so many colleagues, not so many economists. So let me start with a model, because this is a presentation. I know there would not be many economists around, but not so few economists. Maybe that's myself. So this is a very specific presentation for a specific topic, which is the use of affinity factors and the use of adjustment parameters for the economic valuation of ecosystems. This is actually what we do in our group, economic valuation of natural capital, starting from the economic valuation of ecosystem services. So we use standard classifications of ecosystem services, and we use what now might be considered to be a standard methodology for the economic valuation. This is an application over an application, so well, I'll try to be not so specific and not so detailed, but this shows a problem that we usually find when doing some economic valuation using benefit transfer. So instead of doing direct studies, we use data from other studies, and we try to transfer it to our target areas. So this is a proposal for a solution, a proposal of solution for a problem which is commonly found in this sort of exercises. So, well, this is the goal: obtain economic estimates for some region and for some specific industry, which is production of wine. So production of wine is, of course, the source of provision services, but it's also the source of regulation and cultural services. And this is a project which is funded by the Junta of Castilla y León. In Castilla y León, there are, well, these are just part of the wine production areas in the region. Those are the areas which are close to Duero River. There are 2 other regions in the north, and at least another region in the south, which is not so close to the Duero River. But our goal here was to focus specifically on these 4 areas. Probably you heard about Ribera del Duero, maybe not so much, but maybe you heard about Toro. I guess not so much about Rueda, and I guess much less about Arribes, but they all produce fantastic wine. So, for provision and services, well, we use the classification from the CFEA framework. So, but this comes from Millennium Ecosystem Assessment starting in 2005, more or less. This is when it was standardized, even before the final 2005. So, for the provision and services, we use market data. We have a lot of market data for provision and services, maybe except for genetic resources. For regulation and for cultural services, we mainly do benefit transfer, so we do meta-analysis And we try to focus on homogeneous methodologies. So we try to use avoided cost for regulated services, and we try to use travel cost for cultural services. For some cultural services, we could use as well choice experiments, and we could use contingent valuation, but there are not so many studies for these ecosystem services for vineyards. That's why we trust on travel cost for this study. So this is the specific part for affinity factors. What we do for the benefit transfer part. I don't know whether you're familiar with this, but if you try to do meta-regression with papers, with results related to regulating or to cultural services, well, for some ecosystem services, you might have something with a significant, well, with a relevant statistical significance, but the variety is so wide that it's very difficult. We still don't have enough information to parameterize the values. So trying to do meta-regression with this, except with forest we have plenty data, and it's kind of, well, satisfactory to use meta-regression. But for the rest, it's very difficult to find some meta-analysis with meta-regression with significant statistical results. So what we do is we use affinity factors to give more weight to the studies that are closer in primary productivity, closer between the original sites and the target sites. First, we create what we call the naked values. So we remove the part of the value which is due to differences in income and in population density. So we get what we call these naked values, and from these naked values we give more weight to the studies that have been performed in closer in these factors of affinity, which we use pluviosity, we use solar radiation, latitude, altitude, and slope. This is what we use. Those are the most important factors for these ecosystems. Then we get another naked target value, which of course doesn't include the effects of density, population density, and income. And then we get the final value by applying the differences, the different parameters of population density and income. So this is the part for the affinity factors, but then still we have some problems, and those problems which are related to our methodology, to the benefit transfer way. Well, focusing on the last one, primary data is frequently aggregated among all the categories of cultural ecosystem services. So we don't usually have the detail to know what part of the system, of the value, is due to recreation, visual amenity, inspiration. So that's why we might not have enough primary data because we cannot enter into this granularity, and those primary data might not be homogeneous, so we might not be able to differentiate between recreation and visual amenity from the raw data coming from the original sites. So what are we doing? Well, what we do here, you see the results from the application of affinity functions. So according to the distance between the original sites and the target site, we give a different weight to the values, to the naked values that we find for the to be transferred. So we get an estimated value, which is an average, of course, of this, but it's not a simple average, it's a weighted average. So this is for these ecosystem services, 5% of the materials. This is for wild plants used as a source of energy. This is for recreational ecosystem services, which is actually the one which we focus on in this presentation. So the estimated value is 426 with a weighted standard deviation of 524. This might be surprising to you. So how come you are able to publish this? Yes, this is very common. With when we get evaluation of ecosystem services, the standard deviations in many cases are as high as this. So, but again, we have a problem with the aggregation because this 426 is aggregating a lot of information. So we would like to know what is due to the different aspects of culture. So what we have done in this case is to perform a choice experiment. In this experiment, we have asked the people visiting some areas in the wine area of Castilla y León. We have asked them to choose between different scenarios. And what we try to ask them is their evaluation about landscape, knowledge, symbolic value, and recreation in order to split that value for the different parts in the wine area has been analyzed, to split that value into the different categories of value. So this is a regular choice experiment. At the end, we ask them whether they would be willing to pay some surplus over the price of the entrance to some resort. Actually, this is the one that was presented to people at the entrance of the Museo del Vino of Valladolid, which is in a very beautiful castle in Peñafiel. If you don't know it, I would recommend you to visit it. It's in the center of the wine region of Ribera del Duero. Very nice. So the price of the entrance is €6, so we're asking them to pay a small surplus to guarantee some result. So what we find when we do the logic analysis is different parameters about the relative valuation, higher for landscape, second is recreation, third is symbolic, and fourth is knowledge. When we have done this same analysis in different parts of that, those regions I was telling you before that were close to Ribera del Duero, the values are not always the same. For instance, if you go to the region which is close to Portugal, Arribes, you find landscape first, but then the symbolic values are higher than the recreation. In Arribes, the production of wine is traditional. They do it in the ancient caves. Actually, there's a net of caves, which is very nice to visit. Okay. So what we do then is characterize the region according to some factors that provide information about visual amenity, education, spiritual, artistic, and symbolic values or recreation related to the heterogeneity of landscape, published articles in different wine producers or collaboration with research, where we find some celebrations in areas that are related to wine, or where we find areas that are marked to walk through with guides related to bird watching, for instance, things like that. So we created a list of, with all those factors, we created a list of factors, common factors relating each characteristic for each place with, and turning that into an average of 1, so kind of normalizing the factor. And then we were able, we have been able to split the values, the value for recreational value. We have been able to split it into the different characteristics of that value and create a more detailed map, which is this one, coming from that initial figure I told you before. Okay, thank you for your kind attention.
Thank you, Fernando. Now it's time for questions. Julien?
Thank you. So, in order to promote the restoration of ecosystems, do you think that we could also have another financial model like for art, where you have a value that is not linked to a service, but that is linked to the beauty of the biodiversity? Do you think it could work as a market of biodiversity beauty?
Okay, if I understood your question, so art is included, it's an ecosystem service, it's symbolic. So the value of people painting landscape, that's also a value of nature, so it's there. Then when you put a value in that, it's a different thing. And another part of the answer I can think of, the markets are already moving to create assets based on biodiversity. They are doing it now, and that's the reason for the European Union taxonomy. So the market moves very quickly, and the market knows that sooner or later there will be financial assets linked to the protection of biodiversity. So far, the metrics are quite imperfect, not based on consensus yet, but sooner or later they will be regulated, and probably the value of inspiration for art will be there. Was that your question?
Not exactly, but I was thinking of art as itself. You give value to a painting that is not the price of the paint and the oil that you put. Some painting will cost a lot of money and some will not. And it's a way, it's not based on the service they give, but it's based on other factors, and some people are paying crazy money for a painting that someone could find not so beautiful, and so that's just...
Okay. It's a good question because we are surrounded by paintings, so we might wonder what is the value of these paintings. Of course, the short answer in economics is what the market is willing to pay for them. And actually, this is the answer that the United Nations has taken for CI. So it's exchange value, it's not intrinsic value or something like that. And actually, there's no short answer to your question. When we started to do this project, we were wondering what is it that gives value to wine. So can we find the specific parameters of value which are related to nature? And after talking to different groups of experts, we realized of something that probably they knew very well before, which is that the price of wine depends on many factors which are controlled by the market. So what's the reason why some bottle of wine may have a price of 1,000 euros? Is it because it's 10 times better than a 100 euros bottle? And is that 10 times better than a 10 euros wine bottle? For sure it's not. So when you remember that slide when we set the naked value? Our naked value is related to the fact that a lot of value which is put by the market on some assets is not related to the natural characteristic of the asset. So we have to take off the surroundings that have been created by the market and leave it to the natural part of the value, which of course is not always a straight line. This we have to do.
Okay, we have time for one short question, please. If not, we can... Okay.
Make it short. You also include disturbances like erosion, copper accumulation, things that are related to wine production, but are not monitorized maybe until now, or is that part of the research and science?
So the control of disturbances is an ecosystem service. It should be taken into account. If we, for control of disturbances, honestly, I cannot say whether it's in our list, but it should. If it's not, it's because we don't have primary data for it. But it's an ecosystem service in all classifications. Okay.
Christos, please.
Yeah, first of all, I would disagree, about the value of the wine which is probably 10 times higher. I tasted once the Brunello from Tuscany, and I said, would you die without trying this wine? So it's different, but anyway, that was a joke. The point is, do you think that in the long term, the market will work, at the benefit of preservation of biodiversity and conservation of our resources? That's the question. And I guess that you are, the economists are the only one, to bring up an answer. A sincere, please.
Okay. At the beginning of this season, so in October, I was sure that Bernaliz would win the championship, the European championship, and it has not. Now I'm completely sure that the market will work for this. I'm completely sure. The market is moving very quickly. So if you browse about environmental finance over the internet, you will find where the problems are, where the blocks are, but you will see that the financial market is so strong that they are providing solutions continuously. Solutions, come on, the sort of solutions that the market is comfortable with. So I'm sure it will. Okay. And Real Madrid reached the semi-final.
Thank you, Fernando. I think we are going to move to our next speaker, but if we have any additional question, we can continue the discussion at the end of the session. The second speaker is Miroslav Tsvetkov, professor at the Nikola Vaptsarov Naval Academy, who is going to talk about the technologies that are being used by the Spanish and Bulgarian Antarctica bases for ecosystem and habitat monitoring and mapping. Please, Miroslav.
Thank you, Anna. First of all, thank you organizers for invitation to be here and to present our activities in front of you. I am a representative of Bulgarian Naval Academy, which is situated in Varna, Bulgaria. This is on Black Sea, West Black Sea. We are part of Life Watch Bulgaria Node. Our leader in these activities are Agricultural University of Plovdiv. My presentation is not strongly scientific. I will present our capacity, our platforms, our sensors, which we are using for different applied scientific research, and idea to start something together outside Europe to enlarge the Life Watch activities outside of European continent. First of all, my academy is European Space Agency contractor. We start with one project financed by ESA to prepare one software for processing of Sentinel-1 data. This is radar data. All radar theory is put it inside in this software. The project finished with good results. We continue, we verify our results with results and data from 2 European Maritime Safety Agency systems. One of the system is SafeSeaNet, which is responsible for ship traffic monitoring, and second one is CleanSeaNet, which is responsible for ecological domain in European seas. After processing of the data, we include our data in different GIS systems, for example, in Google Earth. We continue to work with Sentinel-2 data. This is multispectral data for different ecological purposes. After we start to using Landsat, MODIS, and commercial system data from Pleiades and Planet. On the slide is shown different digital image processing with respect of ship traffic and ecological purposes. We are using Sentinel-2 data for change detection purposes. On the slide is shown one research connected with this big earthquake in Turkey beginning of February, for change detection of broken or damaged buildings. This index is very useful and for deforestation purposes with respect to ecosystem and biodiversity monitoring. In addition, Sentinel-2 data with respect to water quality monitoring and to eutrophication. We are using data from Sentinel-3 and Sentinel-5B. From Sentinel-3 data we are using especially for marine meteorological purposes and for disaster management. Sorry. On this slide is shown one wildfire. This in black is burned area, in red is burning area. Very easy, we can calculate the damaged areas in the forest, for example. I know from our LifeWatch Eric's Spain that in October will be launched one new satellite for purposes of LifeWatch tasks. This nano satellite will be on with resolution down up to 5 meters actually, and will have revisit time 3 or 4 days. This is very good revisit time and very good resolution, and we are ready to work with this data and to provide some services using this multispectral data. We are using a lot of data from Copernicus services. For example, Copernicus Marine Service. We are using data from Argo buoys and we processing data from historical data for sea currents. For example, one study which is connected to monitoring of drifting object over sea surface using Copernicus Marine Service data for Black Sea especially, where we provide one research that shows how to say transport routes in West Black Sea. Unfortunately, this research was appropriated more than 10 times previous year, tracking these new floating objects coming from Ukraine, from north, and going to Bulgarian beaches or going to south, depends of the season and currents in this season. This is our drone fleet, small drone fleet. We have a drone academy in the Naval Academy, more than 10 flying drones, copter type are more of them, and one wing type, and 2 underwater drones. We are using these drones for drone orthophotogrammetry and for data processing after making this called mosaic. We can see on the picture human footprint, cigarette box, and other objects on the ground. Actually, the drone altitude is 30 meters, and with this good resolution of the camera, we can monitor a very small object on the ground. After we having the photogrammetry, we start to applying different algorithms for edge detection, artificial intelligence, and some algorithms for automatically measurement of the objects and to preparing reports. We are using multispectral sensors on drones with additional RTK base station. This allows to positioning of the drone to be in the maybe 10 centimeters accuracy, This drone especially is equipped with a multispectral camera corresponding to the multispectral cameras of satellites, and this new nano satellite which will be launched in October. Thermal drones. With thermal drones, we are using thermal drones for ecological purposes, oil spills and plastic waste. This thermogram is made by drone with altitude of 100 meters, and the drone measures the temperature between oil spill and water 3 degrees Celsius. Again, multispectral and thermal images. This research we are doing under one ongoing project, which is financed by our country. This is National Research Program for Smart Agriculture. We have RGB image on the. We have a research field with RGB image, NDVI index image, doing with multispectral camera and thermal channel of the same image. When we are using drones, we start to combine video stream with sensor data of the drone. And this is very important point that all object is geo-referenced, and this can be used for further activities, for example, for cleaning some area or something else. Video records from underwater drones. One of the drones is equipped with manipulator, not hand, just manipulator. And using these drones, we can monitor all activities underwater, for plastic waste, biodiversity, and etc. Again, the video records are geo-referenced, and on the picture, on the movie actually, the operator can monitor the sensor data of the drone. We have additional equipment for monitoring of different parameters of the seawater, sensors for measurement of sea currents on the depth, CTD equipment with additional sensors for pH and other important parameters of seawater. Previous year we provide one preliminary research for areas on West Black Sea, actually the Bulgarian part of West Black Sea, which is appropriate for maricultures. From previous year, we are operator of this research vessel. The Academy buy this vessel, and the vessel provides his first mission to Antarctica, 5 months mission from November to end of April. Actually, Bulgaria has a research base in Antarctica, and our research base is very close to the Juan Carlos I. This is one of the Spanish research bases in Antarctica. The distance between these two research bases is 4 kilometers. And this is the end of my presentation, and thank you for your attention.
Thank you very much for this interesting talk. Is there any question from the audience? Please.
Great, Christian Irish from the University of Amsterdam. At one of your slides, you highlighted the recognition of an oil spill, and I'm not sure if I understood correctly, but was this based upon thermal imaging on the identification of the oil spill? And if so, how does that work in practice?
There are two ways to identify oil spill: using radar data or using data from thermal camera. Using radar data, the backscatter signal from the oil spill is very different from the signal from seawater, which is around the oil spill. Using thermal camera, the thermal camera works using the thermal difference between these two different products in general.
Please, we have time for one short question, please.
So thank you. We live in a fairly unusual geopolitical world at the moment. So there are lots of discussion also in our university on should we collaborate with these applications that also military sort of function sometimes. Could be at least dual use is mentioned. Is the stuff coming from China used in your cameras or in your system? So how do you deal with that in your situation? Do you feel freedom to do whatever you like, or are there limits in your opinion at the moment?
We have no limits to use this type of equipment. Actually, drones are DJI drones. This is a very popular Chinese producer, and they come with these sensors integrated. In this domain, the satellite data, for example, or multispectral data, can be used for dual use, but it is depends on resolution of the signal, of the image. These commercial producers, Planet and Pleiades, have a resolution of pixel 50. One pixel is 50 to 50 centimeters. The other producers have a resolution 30 to 30 centimeters. But this is very expensive product. If you want to looking for these small objects over ground or over sea, you should pay a lot of money.
Thank you very much. We have to move to our next speaker, but we can continue the discussion after the session if you like. Thank you. The next speaker is Julien Radoux. He is a scientist at the Catholic University of Louvain, where he's currently working to provide data services for biodiversity researchers within the LifeWatch infrastructure. Julien, please.
Thank you for the introduction. In the beginning of my talk, I will give a little bit of theory about model, but not the species model, but the GIS model, so the models that we use for mapping. And those models are very important to choose because all the process that you will do when you build your model afterwards will depend on the geodata model that you choose first. My teacher, when I was a teenager, always told me that all models are wrong, but that some are useful. So choosing the right model is always a way to not to make things too bad, but none is perfect. Fortunately, we only have 3 models for geographic data. Those are the 3 ones. Then we have spatial object, something that you can really see and represent as a vector, like a point, like a chair. Everyone will agree where is the chair. Then we have the field model. This is more like something that's changing everywhere, like the temperature, or we have the density of grazing in a previous talk. So this is information that's changing everywhere. And then we have the so-called ORCA problem, where you have an olive grove and and you have trees, but where is the limit of the grove? It has to be decided arbitrarily, and better make always the same mistake than doing different mistakes every time. So in this case, we will try to have some rules to define where the boundaries are, and to make those rules consistent so that it can be reused and reproduced and compared to see if there is a change in the definition of this object. So we try to make a partition of the landscape using this spatial region model with arbitrary boundaries and a quantitative description of what's inside the boundaries of delineation. To do that, a common use model is the data cube, but for, as you can see on the examples of prop types, even if prop types are not so natural, they are not always squares. And so if you have a cube with only squares, to define to partition the landscape, you will either lose some resolution or you will lack of concept or of context if it's too coarse. So my goal is to find another way to delineate the spatial units. To do that, we could talk about species habitats, but every species has a different habitat because they need reproduction, they need feeding, and so they will not use the landscape the same way as another species. Some are big, some are small, and so sometimes a tree can be a habitat, or you need the whole forest to be a habitat, or the forest plus the crop field that is next to it. So habitat is not perfect. And we try to find better, and by the definition of ecotope, that was a concept by Ellis, this is a definition that combines the biotope, so the abiotic variables that are in a location, and the land cover that we can observe, because we also want to be able to do the delineation that we represent, because if we target is too ambitious, we will not reach that. So we try to have these ecotope maps, and you see that it's delineated based on the land cover and some abiotic variables. Not all abiotic variables are visible on the map, but fortunately now we have topographic variables that are available nearly everywhere. So we use, in order to delineate automatically those ecotopes, the topographic and the orthophotos, so the geographic information provided by remote sensing. This topography is used, as you can see, with the lines. In black, it's a delineation without taking the topography into account, and in white, it's the ecotope delineation, where when you have a change in color, it means you have a change in slope, and this change in slope will drive a different delineation of the ecotopes. So we get this result, where you see valleys in the forest that are invisible when you only look at the orthophotos. We tested soil properties for those ecotopes, and we had the good pleasure to see that the soil properties were well conserved into better homogeneous into the ecotopes than in the delineation without taking into account the delineation, but only the topography, but only the land cover. So this looks like this for Belgium, where we have very high resolution data, LiDAR 1 meter resolution, orthophotos 25 centimeter resolution. So we have good conditions to do the work. The delineation and the land cover maps are combined to give you the map here, where you have the ecotopes with a label that is only used for your visualization as human, because the machine only looks at the quantitative variables that are stored here, like the proportion of trees, urban and grassland, at different scales around the ecotopes that are computed. All of this is combined with all the information that you can find in Belgium, and where it's very rich with soil maps. So we have more than 100 variables for the 3 million polygons that are composing that. And this was successfully used for modeling habitats of birds, butterfly, plants. Not always the best, sometimes the square works better than the polygons, but the majority of the case we got better species distribution model and species habitat models based on those ecotopes than with a grid of the same area as the average size of the ecotopes. Now it's LifeWatch Europe, so we try to work on Europe. For Europe, we work on the Copernicus data, both the thematic services and the remote sensing data. So it's 10 meter resolution and not 2 meter resolution anymore. But we use the best available sources, and we try to improve the information. If you find errors, please fill this, and so we can improve the map again, so that this map will become better and better instead of having a static map, because the infrastructure aims at building the best map on the long term and not a single project map. So this looks like this, a snapshot on a city that you might know by now, and the place where we've been just a few yesterday. The representation in the ecopatch, so the big ecotopes, is like this. So here it's all red because it's a city, but if you look inside the city, you have a small, you see that you have part of trees. Well, it's good for the bats. We have the presentation of Maria Luisa is there inside with this for the bats that are living in the city. So it means that it's not because it's red that there is no biodiversity. You need to go and look inside those polygons. And again, we also have different information. So we also had a presentation saying, okay, I don't like this map because the trees of olive trees are not forest, but they are crop, or I want them to be crop, or I want them to be forest. Well, for me, the land cover is a polygon containing small trees, and then you can decide based on the land use if it's a forest or if it's agricultural production. But those are two complementary information that needs to be stored in the same database and not making necessarily the difference. So here we have the two labels that are used, and you can change here, here the ecosystem majority, or you can visualize using the prop type, or you can visualize using the land cover type. So you can download the data. For Europe, it's not yet, it will be released end of June, but you can already go to the website, play with the data, and see how it looks like. And comments are welcome.
Thank you. Thank you, Julien, for this interesting talk. Is there any question from the audience?
Thank you for the presentation. What I'm wondering about is, so within Europe, there's quite a large diversity in the resolution of LiDAR data sets. Another project which works on language Eric is working with the Dutch LiDAR data set, which is, I think, 16 points per square meter or so in resolution. And by heart, I think Denmark does something about 20 points per square meter as well. I understood that you are planning on using a European-wide LiDAR data set. So my question is how much does the resolution of the LiDAR data set affect the quality of the map that you can produce out of it? And is it therefore worthwhile to pursue individual data sets of countries rather than one generalized data set?
So for the first question, the resolution of the data sets, for me, at 1 meter resolution, 1 point per square meter resolution is enough for the purpose of building this database, because we want to delineate. So we are not characterizing precisely the vegetation, but we are first trying to delineate it. So for the delineation purpose, 1 point per square meter would be enough, and this maybe now could be available, something we could think of for Europe. 16 points per square meter is not yet something we could get for Europe very soon. So that's the first question. The second one, I forgot it now, but
so because there's differences in resolution, so there is a choice between having a data set which has, I believe, 10 points per square meter or so. If it was worthwhile to pursue these individual data sets of countries to increase the resolution, but I think you've answered it in the first part of the question already, that is not worthwhile, considering that 1 point per square meter is sufficient for delineation purposes.
Yeah, but I don't have the one point per square meter for the whole Europe. So yeah, it's worthwhile if I can collect it from the different countries, then we could reach the ecotop resolution. We could build the ecotop model for Europe. And here, not only the ecopatch model with 10 meter resolution that I showed in the second part of my presentation. So, of course, getting to ecotop and having hierarchical polygons with the ecopatches are about 10 times larger than the ecotops would be very interesting for the whole Europe. But right now, I don't have the data to do it out of Belgium. I have the data to do it in the Netherlands, but yeah, we can talk about it later.
Thank you.
Thank you. We have time for another question.
Yeah, thank you. It's very impressive. A large part of the European biodiversity takes place outside of the continent, so like on the Azores, the Canary Islands, Réunion, which is part of Europe. Is it also part of your work?
No, it's not. I mean, if I start mapping Réunion, I should start all the context, and so it should jump to the global scale then. Yeah, we could go to this, but currently it's only I would say the enlarged continental Europe that has been mapped.
The easy part.
But no, I'm with Belgium, and then we have the whole Europe from northern of Norway to the south of, but not the island of Réunion or the territoire d'outre-mer of France. Yeah.
I was wondering at what point would you decide not to work with ecotopes anymore, because it's sort of a discretization of something that is maybe very continuous. So why don't you work with the raw data more or less and say, well, everything is present. You have all the data, and still I see that it's useful to make nice colors, but is it also useful to keep working with the raw data?
But the raw data, like remote sensing data, you will have some heterogeneity that is not really present. I mean, there was a school of saying that the diversity of the spectral signature that you observe is related to the biodiversity in the area that you observe. But now this has been demonstrated that this is not the case. It's working in some specific case, but most of the case, the heterogeneity of the spectral signature is more due to difference of, it's a bit of shadow because you have trees, or you have a small topographic effect on the water or non-water. So this is not informative for building of most of the models, and having the spatial region allows you to have this context that you don't get if you only look at the pixels. And also you could have pixels that is exactly the same color in a permanent grassland that has been fertilized, and in a deciduous forest where you have a specific tree. So you could find exactly the same spectral signature in both. And if you use this information as the raw information, then you lose the information that we also get from the height that we got because we had some context, that we had temporal information that was used. So yeah, I mean, there are terabytes of data used to build this model, and then it's also a way to have only 12 million objects to use for Europe instead of 130 billion pixels, with the depth of 30 years of observation, 5 images a day, every 5 days with clouds or non-clouds or shadow. So yeah, I mean, I think it helps to have this.
Thank you very much, Julien. Now we move to our next speaker, who is Manuel de Figueiredo Meyer. He is an environmental engineer at the University of Porto, working with the assessment of plant biomass through remote sensing. Please, Manuel, the floor is yours.
Hello everyone, thank you for the opportunity for being here. I'm going to present work that we're doing about herb stratum biomass estimation through ultra-high resolution. So, it's a brief summary, objectives, study area, methodology, we have both ground biomass estimation and further research. So the question that we had was, looking from above, can we identify the biomass of a specific herb stratum community? And we could also always use satellites, airplanes, but we went with unoccupied aircraft system with multispectral image. Red, green, blue, red edge, and near-infrared with a pixel of 2.5 centimeters. So this methodology could be applied to wetlands, prairies, intertidal seagrass, but we went with dunes because it was close to the university. This is the study area, the protected landscape of Seville at Parque Natural do Litoral Norte, and we were studying Campo Bruto de Luís, which is also known as Unha de Gato in Spanish, or hot and fig in English. So this is the study area here, north of Porto, and this is the methodology in a nutshell, right? So we made pilot placements in the fields, and we georeferenced them, and we took the multispectral area images from that. So we made a sample collection of these quadrants. We had the dry weights of the samples, and then we have also vegetation index from the multispectral image. So we try to correlate with empirical regressions this dry weight with the vegetation index. And we used the land cover classification to see if we could identify the Cacobrotus plants. So with these two information here, we could have the total biomass in the area. So the first part of the research, we have the quadrant placements, and I want to show you three things in this image. First of all, don't use a bright white quadrant because there's a lot of reflectance interference. As you can see here, this quadrant was only 2 centimeters wide, but we have like 1, 2, 3, 4 pixels sometimes, and in this one, which is not aligned to the pixels, we have like maybe 5 pixels distance. So it was really important to position the quadrants like in the same position of pixels, north-south oriented. In our case, you can always resample your pixels, but you would be changing a little bit your samples and would give you a lot of work. So the next thing we try, so maybe we don't even need quadrants at all. We have a decent GNSS positioning, and we could do that. But once again, we have some error, 2.5 centimeters error, and image error of 3.5 centimeters, especially in the border. So this is what happened here. The red square was probably where we got our sample, and the green square were where the GNSS marked. So we have an error here, and we'll be considering bare soil, which was really bad. So we went to another approach. We used a bigger quadrant this time with 50 centimeters, and we only took the image with this quadrant. And after that, we put the smaller quadrants that did not appear in the image, so we could know exactly where the samples were coming from without the interference in the reflections of the quadrants. So we took 30 samples using this strategy, and we have this specificity of Cacobrotus, which is it grows in layers. You have a green layer upper side, and underneath this green layer, when you take it out, there's a dry layer. So we separate these two because the green layer could be seen from above, and the dry layer could not. So we dried and weighed all these samples. And the next thing, we want to see if there were any relation between the dry weight of the green samples and the vegetation index in the image. So to do that, we tested 16 vegetation index. I mean, we wanted to find the best one, so it could be any of them, and DVI, of course, would have some predictions. But the best one, then we plotted, and we have, well, in DVI here, you can see it's decent, and we have some really bad ones. But we used these 3 regression models to see if there was any relation. And the best one was the renormalized differentiate vegetation index, which is basically the NDVI with root square underneath it. So we had an R square of 0.87 and a normalized root mean square error of 0.09. After that, there was the big question, can the classification models identify the Capobrotus vegetation? So we tested many different algorithms, but the best one was the random forest algorithm, followed up by a sieve filter to consider the surrounding pixels. So we got a user accuracy of Capobrotus of 0.8, producer accuracy of 0.9, an F1 score of 0.85, and an overall accuracy of 0.89, which is pretty acceptable for our classification. You can see here the image is a little bit dark, but this is the classification. The dark one is Capobrotus. So this is the random forest without considering the surrounding pixels, and we use the sieve filter to consider the surrounding pixels to have a more homogeneous image, which represent the reality a little bit better. So we were applying sieve filter with progressive strange to see which one was better, because once you use a filter, you lose some information, but at the same time, you consider the surrounding pixels and you can gain some accuracy, which was always a balance that we had to make. Then we have the regression models, we have the Capobrotus classification, and we could try to estimate the total biomass in the area. So the estimation, the total dry weight of Capobrotus was a summatory of dry weight of green parts from each pixels, which was a function of the vegetation index that we had before. We also have to consider that the dry weight of green parts is just a small part of the above ground biomass. So we have the dry weights. We have to consider the relationship between the dry weight of green and brown parts, which did not present any correlation that could be used with empirical regression. So we made an average of 3:1 from the data. And the dry weights and wet weights of green parts, which were pretty linear, 1 to 10, and the dry weights and wet weights of brown parts, which are also linear. So to validate the accuracy of the results, we could use many different techniques. The first we had 30 samples. We used 25 for classification and 5 for validation, which gave us a 100% user accuracy and producer accuracy. And anyone who works with classification knows that it's not a good result because it's completely unreal. So we could use a K-fold classification, but that takes a lot of process and might still include the 100% user accuracy, which was also not good for us. And we could also create validation areas, but they would be positioned at some point, which takes a little bit of the randomness out of the validation, which is not the ideal, but could be used. But the sieve filter makes some areas really homogeneous. So once again, we could have the problem that the homogeneous areas were really generating overrated accuracy. Like you can see the sieve filter here. There are some really homogeneous areas where we would probably place the validation squares. So we went to random areas. We selected 600 pixel size areas, randomly selected through the image, which 518 could be securely identified. And then we have a 95% confidence interval of 10% of the area, which of course, once again, as Julien said, it's an estimation, right? No model is perfect. And well, that was basically it for the methodology. And we have some further research to do. So can the classification algorithms always differentiate a species from other species? Like this is true for Capobrotus, but would it be true for other species? Will the above ground biomass and vegetation index work with these other species? Maybe they don't have a good relation. Is there any specificity of the plants that needs to be considered? I mean, Capobrotus grows in layers. Maybe if we are investigating other plants, we should consider other specificity of it. So it's important to understand which plant we are researching. And the next research that we're doing right now is remake all the process with bigger pixels from airplane data. So we can monitor a larger area of the coast and understand how is the invasion of Carpobrutus in north of Portugal. So how much accuracy is lost with it? Of course, you have bigger pixels, you probably will lose some accuracy because of the size of the plant. But there's something that still needs some research, and we are processing the images right now. So that was basically it. Thank you for the attention.
Thank you very much, Manuel, for this interesting method to estimate biomass of the Earth stratum. Is there any question from the audience? Please, Julien.
Yeah, so with the drone data, we see that there is a lot of fuzziness. Did you try to remove it using some deconvolution filter or using the information about the fuzziness in the process?
We use the C filter, which actually gets some here. We use a C filter that's considered the most predominant pixels around a threshold area of a limited amount of pixels, and we started with 1 pixel. So if you have one lonely pixel there with predominant class surrounding it, it would be changed to this predominant class.
Yes, but the C filter is applied on the result of the classification.
Yes.
I was wondering if you tried to use to diffusify the image before applying.
No, we did not do that. But it would be interesting to test, definitely.
Thank you for interesting presentation. I have only one question. What is the confidence level of GNSS accuracy?
The confidence level of GNSS accuracy. I don't have the information of the confidence level. We use the
We'll discuss later.
What?
We'll discuss later if you want.
Yeah, it would be very interesting. We use the error that comes with the Porto mosaic production software, so it comes with an error attached to the information from the GNSS. So that's the error that we're using, but we'll have to discuss it.
Thank you very much, Manuel. I would like to welcome our next speaker, Joaquín Osorio Arjona. He is a postdoctoral researcher at the University of Seville. He is going to present a review of voluntary geographic information projects for ecosystem and habitat mapping.
Thank you very much. Okay, I'm Joaquín Osorio, and we are going to use the geographical information from a different approach. And then this is a project made by the group in GES, whose leader is Berenberga Mateos. And the group is part of the INDALO project, Scientific Infrastructures for the Monitoring and Adaptation to Global Change in Andalusia. Geographic, that is since 19th, and we have used spatial data infrastructures for the use of our geographical data, right? And geographical data derived from institutions which are rich in metadata, but the rise of new technologies of information and communication are giving us a new opportunity to give vast quantities of data in real time, the famous case of big data. In this context, we have the voluntary geographical information, which can be a very important, very valuable tool to getting information of ecosystems, of infrastructures that spatial that infrastructures and the institutions won't be able to get us. Just to simplify the definition, voluntary geographic information is tools to create, assemble, and disseminate geographical data provided voluntarily by individuals. And the most famous example we can have in mind is OpenSeaMap. Voluntary geographical information can be used as a georeference type of citizen science, and it has and there is an established tradition of volunteers contributing with their own geographical information for the analysis of a situation in an ecosystem. More or less, the characteristics of each platform is that voluntary geographical information, when we can give us a lot of information by voluntary sensors, that information usually doesn't have metadata, because we are harnessing information with our mobile phones, and many times is by volunteer citizens which have specific information in the UC and we are analyzing. However, spatial data infrastructures, since they are institutional initiatives, they are reaching metadata. Textual information blocks have a great potential for combination. To try to combine the institutional structure, the reaching metadata of spatial data infrastructures, with the flexibility and dynamicity of the volunteer geographic information. Okay. The study we have made is to identify volunteer geographical information projects related with ecosystem and habitat mapping. And we have made a first description of the main features, including the promoting entity, the scope, and the number of records, the spatial scales, the use of technology, and the characteristics associated with voluntary geographical information and the characteristics associated with spatial data infrastructures. To see if these maps can have both voluntary geographical information and spatial data infrastructures characteristics. And the next two diapositives are the 23 projects we have found, which are divided in three categories. A report of environmental conflicts, for example, report conflicts related with use of water in Andalusia, environmental crisis, for example, problems that have been suffering, our population has suffered by typhoons, by natural disasters, and citizen science related with environmental projects. These are usually projects made by universities, which, for example, institutions, for example, in NASA, which has created a map for map earthquakes, landslides. We have found 22 maps, and 5 web maps were focused on reporting environmental conflicts, which the source is diverse, academic, and non-governmental organizations. The web maps focused on reporting environmental crises have come mainly from non-governmental organizations, and the 13 web maps focusing on citizen science applied to environmental projects have come from academic sources. This last category is the one which have more records. In some cases, it takes dozens of records because it has more support. It's more easy for academic projects to have team of volunteers which knowledge of what to map. And web map that reports by environmental conflicts tend to be created using open source tools. And open source tools are technologies that everybody can use. It doesn't have a proprietary license. However, the citizens project, the citizens web maps usually are the opposite. They use property license. We have found that only two web maps allow sharing data using open source service. Only 8 maps have metadata. Only 3 web maps have spatial, temporal, or thematic data search tools. And only 5 maps allow the download of data in geographical format like zip file. And the main way to download data is to forms supervised by maintenance teams. And only 12 of the analyzed web maps allow the download of data. The main data download format is XML or CSV files. Citizen size web maps present a low degree of accessibility and a more complex interaction. And most of them require a prior registration and user verification process. In conclusion, the listing was to be done to integrate both voluntary geographical information and spatial data infrastructure projects. There have been 2 types of projects. The ones who have spatial data infrastructure has a priority, and the ones whose main goal is to report directly a conflict, a problem. There is a complex range of highly differentiated functions, and we must continue working on technological solutions that favor sharing, viewing, consulting, and reusing data for habitat mapping. And a need for building institutional knowledge. And thank you very much for your attention.
Thank you very much, Joaquin, for this interesting presentation. Is there any question from the audience? Please.
Thank you for inviting this presentation. I was wondering if you have also considered the sustainability of these systems, because volunteers need somehow some kind of feedback. So part of this feedback could be if they contribute, they can be in a knowledge list or something like that, but also you can provide a kind of product that is useful for them.
Yes, usually a characteristic of the voluntary geographic information, a possibility that is given, is the possibility of giving feedback to their own information. The users and data point with data, and other users, not only the owners of the map, but other users can evaluate the information, give a score. When we have analyzed things and we have had it in the main paper we are working, there are still few maps that are in this tool, and about sustainability, and precisely because we are analyzing web maps, almost all web maps that we can access are mean that they are accessible and currently accessible. Maps that are sustainable are usually disconnected from internet. They stop working, the servers stop working, and we can't access them. For example, the Usahiri platform, we have stopped working in 2020, and the web maps are hosted in that platform. We have lost that information.
Thank you. We are running out of time, so I suggest to move to our last talk of the day. Thank you, Juan. I would like to introduce Jorge Prieto Rubio, Jorge is a researcher at the Estación Experimental del Zaidín, located in Granada, and he's going to talk about biodiversity monitoring through ecological interaction networks.
Thank you for the presentation. Nice to see you, and hi everyone. It is the last talk of the day, so I hope you enjoy. I would like to introduce on biodiversity monitoring by using ecological interaction networks, paying attention on two types of interaction networks that are occurring in forests, particularly plant canopy recruit and plant mycorrhizal fungi interactions. Ecological interactions are components of biodiversity, since it allows to estimate the interdependencies of a species integrating a community, and even the possibility of a species coexistence in a given habitat. Additionally, these ecological interactions through analytical approaches can estimate several ecosystem services provided by these interactions. We can find some examples in this picture, such as pollination networks, all the mutualistic interactions occurring in natural systems, plant interaction networks, such as facilitation that promote, for example, forest regeneration, and others related to antagonistic ones, such as those occurring in agroecosystems and also in forests. Biodiversity monitoring is traditionally based on a species richness basis, since they individually count species in the different habitats of a study. But when including an interaction network-based approach, we can include not only this species richness information, but also we can include the interactions that are occurring within the species integrating that community, and to solve different questions such as what happened whether this species goes to extinction, and how this extinction determines the possible effects into the others. All these ecological interactions are mainly defined by positive to neutral to negative effects on these interacting species. Particularly in forests, this approach are revealing new insights on forest community structure and dynamics under different environmental contexts. In the project we are carrying out in Granada, the working package that we are carrying on there, we're focusing on two types of ecological interaction networks. The plant canopy recruit interactions, that is mainly based on who recruits under whom, and we can try solving questions such as how is the successional status of forests, and how will respond this forest to the possibility of a species extinction with our communities. And also plant mycorrhizal fungi interactions. This is a symbiosis between plants and particular soil fungal lineages, and that mainly consists in a nutrient exchange in the root system. These fungi colonize the roots of the host plants and promote a nutrient exchange. The host plant provide carbohydrates from the photosynthesis to the fungal symbionts, whilst the fungi supply water, phosphorus, nitrogen, and micronutrients to the plants. To conduct this study within the SUMHA project, we selected all the red natura areas located in the Andalusian region, which are they're small figures, small triangles that are marked in red. And we have selected different vegetation communities that are defined by the Habitats Directive, and for which we can define environmental gradients and also different human impact extent across all these areas that are now under protection. Briefly, the sampling design in these regions consists because we are carrying out, actually, we continue to doing sampling, and is basically by delimiting a total cover of 1 hectare per study area, and we randomly select 20 plots per study area, and we sample all the plant canopy and plant recruits that not only are occurring under the crown of the canopy plants, but also those recruits that are growing out of the canopy influence. With all this information, we have recorded an interaction frequency based on how and to what extent the different plant species are interacting by delimiting canopy plant species with the recruits growing under the crown. With all this information we have recorded across all these red natura areas, a total of 223 plant taxa belonging to 110 genera and 38 families. With this information, we are starting to infer recruitment networks at which we can detect which plant is having a canopy service onto the potential recruits. And we have account nowadays more than 100,000 recruiting individuals involving more than 5,000 paired interactions across all the areas studied. Now, the next part is to characterize these plant mycorrhizal fungi interactions. We focused at the first point on the canopy plants having this type of symbiotic interactions in the roots. And from the list that possible mycorrhizal interactions, we found that those that are related to arbuscular mycorrhiza was the dominant in the forest that we sample. We follow this classification on previously databases that are established, and also followed by these dominant arbuscular mycorrhizal interactions, we found that species that have the possibility to simultaneously show ectomycorrhiza and arbuscular mycorrhiza was the next most dominant interaction. Following to these results, we found a phylogenetic conservatism of these plant mycorrhizal fungi interactions, since we found that closely relative species tend to show the similar mycorrhizal status. Now, the last part of our pool of biodiversity monitoring in this red natura area, we selected one of these study sites to characterize which fungal taxa are composing these mycorrhizal statues in the study sites. And by characterizing the fungal communities in the roots, we could delineate differences of the mycorrhizal fungi number across the different plant families that we have recorded in the different study sites. Moreover, we could record a total amount of 38 families related to ectomycorrhizal fungi and 8 families of arbuscular mycorrhizal fungi. All this information, with the exception of all of these related to the characterization of fungal communities, are now published in a public repository, which is Digital Physique, but will be transferred in GBIF, a platform for the next weeks or months, maybe, I hope. And well, thank you for all the financial aid provided to develop this project, and thank you for attending.
Thank you very much, Jorge, for this interesting presentation. Do we have any questions from the audience? I know it's late and we are all very tired.
Time to rest.
Okay. Please, Julien.
I'm not an ecologist, so I'm wondering if predation is considered as an interaction, because I didn't see the picture of predation.
How do we establish this prediction?
No, predators. Is being a predator considered as an interaction between the species? So if a cat eats a mouse.
Yeah, this is another part, but which is not integrated in the SUMHA project.
Okay.
And we are integrating. We are also integrating herbivores and other biotic components of these possible interactions in order to create a multi-layer networks in the future. We are now elaborating these algorithms.
Thank you very much. So if there is no more questions, before we close the session, I would like to thank all the presenters, all the speakers for their interesting presentations, and all the people from the audience for being here today. Thank you very much.